Okay, let's dive into this. Here's a quick look at our roadmap. We'll be exploring the global lupus mystery, the cumulative genetic load, ancestry-specific genetic triggers, the C1q deficiency factor, and finally wrapping up with personalized medicine.
Section 1. The Global Lupus Mystery. Demographic Disparities.
So family and twin studies tell us that genetics play a massive role here, accounting for anywhere between 43 to 66% of lupus heritability. But that brings up a really crucial question for us. Why does lupus strike so differently across the globe? I mean, why do certain groups face significantly higher risks of severe, life-altering complications, like end-stage kidney disease, while others just don't?
And this brilliantly illustrates exactly what we're talking about. The incidence of lupus nephritis reveals some massive disparities globally. In white populations, the incidence is about 20.3%. But look at what happens as we move to Asian populations. It jumps up to 36.8%. African populations face a nearly 40% incidence, and for Hispanic populations, it's almost 50%. That is a staggering disparity in clinical outcomes.
What makes these statistics even more compelling is that they literally do not change significantly when these populations migrate. For example, an individual of African ancestry who moves to North America or Europe still carries that higher risk of developing SLE compared to someone of European ancestry. So the environment changes, but the risk persists. And that points directly to an inherent genetic link, which perfectly sets up our very first suspect in this mystery.
Section 2. The cumulative genetic load, the quantitative model.
In the scientific community, this first framework is often referred to as the quantitative model of lupus susceptibility. To understand the polygenic risk score, it helps to think of it like a backpack filling up with small rocks. A single, tiny genetic variant, one little rock, isn't going to trigger the disease. But as you accumulate these variants, your backpack gets heavier and heavier until that cumulative genetic load eventually snaps the straps and breaches your immune system's tolerance threshold. So in this model, you don't get lupus because of one single bad gene. You get it because you inherited way too many minor genetic risk variants that just collectively overloaded the system.
And just how many of these little variants are we talking about? Over 100. Genome-Wide Association Studies, or GWAS, have actually discovered more than 100 distinct risk loci associated with SLE. It's a highly complex, multi-gene issue. No single allele is individually essential for the disease to trigger, but together they tip the scales.
Crucially, this quantitative model helps explain the severity of the disease. Research shows that developing lupus nephritis isn't tied to one specific gene you inherited. Rather, its severity positively correlates with the total accumulation, the total weight, of those risk variants. A higher cumulative genetic risk score generally means more severe disease, which gives us one really solid potential explanation for those massive disparities we saw a few minutes ago.
Section 3. Ancestry-specific genetic triggers, the qualitative model.
But what if we look at a counterargument? What if it's not just a cumulative accumulation of tiny variants? What if the risk is driven by specific, high-impact genetic anomalies, massive boulders in our backpack, rather than small rocks? We're going to compare two completely distinct, ancestry-specific qualitative paths to lupus, the BAF variant found in Sardinians and the TNSRF13B and TLR7 variants found in Asian cohorts.
Let's look at the Sardinian anomaly first, because this is wild. There's an insertion-deletion variant in the TNS-SF13 gene called BAF-VAR. It basically forces the body to ramp up levels of a protein called soluble BAF, which intensely upregulates humoral immunity. This leads to highly active SLE and frequent lupus nephritis. But here is the truly fascinating part. Scientists believe this genetic variant is actually an evolutionary adaptation to malaria. Malaria was historically prevalent in Sardinia, so this variant protected them in the past, but it accidentally ramps up autoimmune risk today. And get this specific variant is practically absent in African and Asian populations.
Now, what's really interesting about this slide is that Asian populations arrive at SLE through completely different genetic mechanisms. Instead of the Sardinian BAF variant, patients of Chinese origin often show a missense variant in TNSRSF13B, which encodes a BAF receptor called TACHI. On top of that, meta-analyses show that specific variants in the toll-like receptor 7 gene, specifically RS3583839 and RS179101, significantly increase SLE susceptibility in Asian and Arab populations. These variants alter immune system function and pathogen recognition in entirely different ways, creating a whole different genetic pathway to the exact same disease.
Section 4. The C1q deficiency factor,
consanguinity, and rare defects.
Okay, let's look at an even more extreme end of this qualitative model, over 80%. Just take a second to process that staggering number. If you are born with a genetic defect in the early complement proteins of your immune system, your risk of developing SLE isn't just slightly elevated, it is almost guaranteed.
To ground this directly in the source material, the research states, quote, "A genetic defect in any of the early complement proteins of the classical complement pathway is associated with a high risk, greater than 80%, of developing SLE." This is such a stark contrast to that polygenic model we talked about earlier, where hundreds of genes have tiny effects. Here, literally one broken mechanism completely dictates the outcome.
And the biology here is actually quite straightforward. The first complement protein C1q is basically the cleanup crew for your body. It is absolutely essential for clearing out dead cells and degraded chromatin. When you lack C1q, that cellular debris just sits there. Your body essentially panics, loses tolerance to its own endogenous antigens like double-stranded DNA, and launches a massive autoimmune inflammatory attack. This usually leads to very early and very severe kidney damage.
The data also shows us where these rare defects are most concentrated globally. Homozygous mutations, like this C1q deficiency, appear more frequently in populations where there are higher rates of consanguinity. Studies have noted these rare defects disproportionately in specific cohorts from regions like the Middle East and North Africa, where consanguinity has historically been more documented.
Section 5. Personalized medicine for lupus, synthesizing the models.
So how do we synthesize these two frameworks? Well, the reality of this genetic mystery is that it's absolutely not an either-or debate. The global lupus landscape is actively shaped by both models. Some patients get sick because of a massive quantitative load, that accumulation of over 100 minor risk loci breaching their tolerance thresholds, while other patients develop the disease because of qualitative alleles, those specific ancestry-linked variants like the Sardinian BATH-VAR or the Asian TLR7 variants that hijack distinct biological pathways.
So, the crucial point here is that acknowledging these distinct genetic paths is the only real way forward for targeted, personalized medicine. Because Asian cohorts have that specific TACI receptor variant, researchers are proposing that TACI blockers might actually become the preferred therapy for them. However, and this is a big however, we have a massive blind spot right now. African and Hispanic populations who have the highest rates of severe lupus are severely underrepresented in current genomic studies. We desperately need larger, diverse global studies to isolate the true causal genes across all ancestries.
Which leaves us with this final thought for you to ponder today. As we finally begin to map out these incredibly diverse genetic pathways, how will this redefine who gets which treatments in the future of rheumatology? Think about it. If we can solve this genetic puzzle entirely, we won't just be treating the broad symptoms of lupus anymore, we'll be targeting the exact personalized genetic cause of it. Thank you so much for joining me on this explainer. Keep questioning the data, and I'll see you next time.